Great. And the next speaker is Daniel Sens from Helmholtz Munich.
Hello, I'm Daniel, and I'm presenting GEMCONT, Genetics-Based Multimodal Contrastive Learning. Genome-wide association studies have identified thousands of genetic variants associated with complex diseases, yet for most of these we still lack an explanation of their effect on human tissue. Medical images can help us address this challenge because they capture the downstream effect of these variants on human tissue. Because these images are often very high-dimensional, we need robust deep learning methods that can capture disease-relevant features and enable downstream genetic analysis. Existing supervised approaches often require extensive annotations, whereas unsupervised ones can capture scanner artifacts that can overshadow the disease-relevant signal. In GEMCONT, we instead use disease variants, pair them with the corresponding medical image, and learn a shared latent space similar to CLIP. However, genetic variants exert very small, mostly additive effects on a phenotype, so treating them like information-rich text or images would fail without modification. So how do we address this in GEMCONT? In summary, GEMCONT learns image embeddings by embedding or protecting disease variants and medical images into a shared latent space to then apply the CLIP loss. Because genetic effects are mostly additive, we drop the encoder for the genetic branch and use just a single linear layer as the genetic projector. And because during training we only use variants that are strongly associated with a target disease, we ensure that the image embeddings capture features that correlate with the clinical outcome. And we show that this works by applying our method to UK Biobank spirometry for volume curves, which are routinely used by clinicians to diagnose asthma. And here we use GEMCONT to co-embed these curves together with asthma disease variants, and we learn image representations guided only by genetics. And even if we don't use labels during training, this way we see that the resulting image embeddings outperform unsupervised baselines in terms of genetic signal recovery and asthma prediction. And if you'd like to hear more about our other experiments, please come visit my poster during the session. Thank you.
Thank you, Daniel.